This project profiles a tomato leaf disease dataset published on Kaggle under the slug YOLO-annotated or tomato leaf disease. The data carries YOLO text format, bounding box annotations across 10 disease classes, with roughly 15,000 labeled images and 16,913 total boxes. No detector was trained in this pass. The delivered artifact is a reproducible dataset, Triage-Layer, whose purpose is to condition any future training experiment on specific, documented facts about the data. The framing is deliberate. A metadata-first pass, honestly scoped, is a stronger foundation than a rushed training run with under-reported settings. Over the next 10 slides, the presentation walks through the agricultural stakes, the dataset structure, the 2-count scopes maintained without smoothing, the class imbalance finding that anchors the work, the limitations documented from Barbato, 2018, and Ramkiran et al., 2018, and a human-in-the-loop recommendation that any downstream detector must sit inside. Nothing in the narration requires a trained model, and nothing in the narration claims one. Foliar tomato disease causes yield losses of 20 to 40% for smallholder farmers across sub-Saharan Africa, South Asia, and Latin America. Mohanty et al., 2016. That is the operational context for any leaf disease detector. A grower walking rows by eye can spend most of the morning on healthy plants before reaching an infected patch, and by then the early intervention window has often closed. A detector that localizes suspicious regions on leaf images tightens the search radius before a human reviewer ever touches a plant. I want to be explicit about the boundary. The detector flags candidates. An agronomist or experienced farmer confirms the diagnosis and decides the management response. The tool is a triage layer, not diagnosis. The error costs run in both directions. A false positive triggers unnecessary pesticide application, with cost and environmental burden that smallholder budgets cannot absorb. A false negative lets disease spread unchecked across neighboring plants. Both failure modes matter, and both shape the evaluation plan that a future training run will have to satisfy. The dataset was pulled from the Kaggle repository YOLO-annotated or tomato leaf disease, YOLO-annotated ND. 2-count scopes are maintained without smoothing because reconciling them is part of the deliverable. The Kaggle metadata summary reports 15,225 training images. The local file system, after download and verification, contains 15,209 total images split as 12, 100. 60 images documented rather than rounded away. The most plausible explanation is a download sync artifact, not annotation corruption, and resolving the gap is the first step listed. The annotation format is YOLO text standard. Each image carries a paired label file, whose lines contain a class index followed by 4 normalized bounding box, coordinates expressed as fractions of image dimensions. The full annotation volume is 16,913 boxes, spanning 10 disease classes. The presence of bounding boxes rather than image-level tags matters for scope. This is a localization problem, and a future model must identify where on the leaf a diseased region sits, not only which disease the image contains. I want to be direct about what this pass did and did not deliver, because scope honesty is the point of the artifact. The pass audited the Kaggle metadata, inventoried the local file system across both splits, parsed a representative sample of YOLO annotation files to confirm the coordinate format, generated class distribution and annotation density figures, reconciled the 16 image count discrepancy into a documented gap rather than a hidden one, and recorded the rationale behind each scope decision. Those are reproducible deliverables. A future engineer can verify independently against the repository. The pass did not run a YOLO training loop, did not produce a train detector, and therefore carries no map, precision, or recall values to present. The constraint was practical. Fine-tuning a YOLO-8 checkpoint on roughly 12,000 images for 50 epochs requires GPU access that was not available in the course timeline. A rushed CPU-bound training run with early stopping would produce numbers that look like results and mean less. Documenting the absence is the defensible scientific move, and it leaves the next phase on solid footing. The class distribution is the most concrete finding in this pass and carries direct implications for how a future training run must be configured. The 10 classes span tomato, BacterialSpot at 21 images, tomato, early blight at 190, tomato healthy at 663, tomato lat blight at 20, tomato leaf mold at 142, tomato MosaicVirus at 428, tomato spot at 678, tomato target spot at 678. The ratio between the largest class, BacterialSpot, and the smallest class, MosaicVirus, is 4.7 to 1. That imbalance is not unusual for agricultural datasets, since rarer diseases are harder to photograph and harder to annotate in volume. But it creates a training risk a future detector must correct for. Without weighted loss or targeted augmentation, a model trained on this distribution will encounter BacterialSpot examples almost 5 times more often than MosaicVirus examples, and recall on the rare class will degrade disproportionately. The dataset does not need to be rebalanced physically. What the imbalance demands is disciplined, per-class reporting, and weighted training choices named before the first epoch begins. The figure on screen ranks the 10 classes by training image count with the 522 mean line drawn through the distribution for reference. 4 classes sit at or above 2,000 images, led by BacterialSpot at 2.1, late blight at 2.0, and yellow leaf curlvirus at 2.0. with Septorial-Spot close behind at 190. 3 classes sit near the mean, namely spider mites at 678, healthy at 663, and target spot at 404. 3 classes fall well below the mean and define the minority tail, early blight at 190, leaf mold at 142, and MosaicVirus at 428. It holds less than a third of the mean class size and less than a quarter of the largest class. For a future detector, this is the first class to watch during training, and any per-class evaluation report that omits MosaicVirus-specific recall is hiding the most likely failure mode before it shows up in deployment. The 2-panel figure pairs class size against annotation density, and the combination is where the operational headline lives. The left panel repeats the ranked class counts. The right panel shows boxes per image for each class. BacterialSpot 1.08, early blight 1.12, healthy 1.19, lat blight 1.6, leaf mold 1.99, MosaicVirus 1.48, Septorial-Spot 1.5, spider mites 1, target spot 1, MosaicVirus. That is a doubly challenging class for any detector. Fewer examples to learn from, and each example contains more small regions the model has to localize correctly. A class with 428 images at 1.48 boxes per image is working against the imbalance from 2 directions at once. The practical consequence is that a future training configuration should weight MosaicVirus loss more heavily, augment its sample aggressively, and report its per-class recall and map at 0.5 I alongside the aggregate, not subordinate to it. This slide covers limitations honestly, and I shift to first person to mark the interpretive move. 2 published findings bound my confidence in what a detector trained only on this Kaggle set would do in the field. Barbado, 2018, shows that small dataset plant disease models do not transfer cleanly across growing regions, camera equipment, or disease stages, which means generalization beyond the Kaggle conditions is an open question until a field trial says otherwise. Ramcharan et al. 2017 found that human-in-the-loop validation is essential for cassava disease deployment, and I take that conclusion as binding here too. Error asymmetry matters in both directions. A false positive on a healthy leaf triggers pesticide application with cost and environmental burden farmers in smallholder systems cannot absorb. A false negative lets a diseased region spread. A false negative lets a diseased region. MosaicVirus, with its 428 images and 1.48 boxes per image, is the class most likely to generate false negatives from an undertrained model, which is the failure mode with the highest downstream consequence for crop spread. Naming the limitation this sharply is what makes the recommendation defensible. My recommendation is that the next phase commit to a YOLO-8 baseline, fine-tuned for 50 epochs on the 12, 168 image training split, with evaluation on the 3, 41 image validation split. Redman and Farhadi, 2018. Tu et al., 2019. Tu et al., 2019. Per class precision and per class recall accompany the map table. Particular emphasis goes to MosaicVirus and leaf mold as the two minority classes, because an aggregate number will look acceptable while hiding the specific recall failures that matter most in the field. The 16-image discrepancy between Kaggle metadata and the local file system is resolved before the training run begins, not after. The deployment target remains a human-in-the-loop pipeline. An image enters the system, the detector runs inference, and returns boxes with class and confidence. An agronomist reviews the flagged regions in plant and field context, and only then does management action follow. The order is image, inference, review, response, and compressing that order is where the ethical boundary breaks. The repository holds the Kaggle metadata audit, the file system inventory, the 16-image reconciliation, the per-class image and box counts with densities, the pipeline diagram, and the figures used across this presentation. The two artifacts that carry the operational headlines are the ranked class count chart with its 1,522 mean line, and the two-panel comparison pairing class size with annotation density and flagging MosaicVirus. Together they document a 4.7-to-1 imbalance, and the double challenge finding that any future training run must design around. Nothing in the deliverable requires a trained model to be useful, and nothing claims one. The metadata-first framing was the honest choice given the GPU constraint, and it leaves a clean foundation for the next engineer to pick up. If I were handing this project off tomorrow, the reader would start with the reconciliation table, move to the class count and density panels, and confirm the YOLO-8's plan against the limits noted by Barbado, 2018, and Ramcharan et al., 2017.